Wednesday, when I talk about essays, maybe one possible essays from the virology, you'll have watched the video, so when I say what's a possible essay, it'll at least make some connection in your head. You'll go, oh yeah, I just listened to those videos, I know what she's talking about, I know where in the notes that is. So I'm just pleading with you. I know you have other classes, and you have jobs, and you have kids, and all that kind of stuff. So somehow or other, try to watch those virology videos, and try to be on time on Wednesday and ready to rock and roll and hear about the essays. Okay. Okay. So what I want to start with is somebody. This is somebody's really nice thing that was attached.
Fine.
Is that yours?
Yeah,
Yeah. What happens?
Floppy.
Yeah. These things fall off. Sorry.
No.
Okay. Thank you, Emily. So, by the way, this is only I gave you 10 minutes to look at this. If this is not enough time for you to look at a quiz or a lecture exam, come to office hours if you can. Okay, or maybe after lab you can ask me things that didn't make sense to you. Because I know a lot of you right after class have to run off to jobs and kids and stuff, so you might not be able to look at it. So I always want to make sure you at least can have a quick look at it. And you can see if maybe, I always say this, and students don't always pay attention, but I make mistakes sometimes. Not that often, but sometimes when I'm recording it in Canvas, or maybe I don't notice an erasure mistake. When you erase something and it's not completely erased, and Scantron marks it as wrong. So if nothing else, you should look at your Scantrons really briefly for that sort of thing. Okay, so because you never know, point here or two might help. So speaking of a point here or two, you might have noticed we are having one of two speakers on Fridays that would be useful to go to. So I sent out a little announcement about this Friday at 10 o'clock in SM 211. That's the sort of large lecture, like right over there. We have a lady that is the director of our MLT program here. So the MLT program is a certificate program to work essentially as a lab assistant in a clinical lab. So that's not the only job, though, that is available in the clinical lab setting. So there are laboratory professionals with bachelor's degrees, with master's degrees, with other things in public health and in other specialties. So she's going to come on Friday and talk a little bit about some of those opportunities. If you can come, please do. If you can't, don't worry about it. There are other extra credit opportunities, but it's just one of extra credit opportunities that we allow you to do. There'll be another one a little later in the semester, which will be kind of interesting on the microbiome. So bottom line, if you can come on Friday, please do. It is this Friday, not May 8th. Thank you for a couple of people. Notice the first flyer I sent out said May 8th. No. It's March, or said May 6th. Anyway, it had the wrong date. This Friday, which is March 6th. So, anyway, come if you can. I think it'll be like an hour talk, basically, and then you just have to go and write me a little brief review. By a brief review, I mean, imagine you go see a movie. Okay? And you come out of the movie and you say, I like the movie because of X, Y, Z. I learned this in this talk, X, Y, Z. I didn't like this part. I thought this was boring. I like this. I thought this was, that's what I mean by a review. Just like you would do if you were telling, one of your classmates about what was talked about in the talk on Friday. So it has to be literally half-page handwritten. No big deal. Just tell me a little bit about what you thought. And those, we actually take those seriously because we read them and it's like, you know what, everybody hated this. We're not asking the speaker back. I mean, literally, we use it for that. Okay, or it's like, wow, students were interested in this. They learned stuff they didn't know. So we do actually read those over. And it's worth 5.6 credit, can't hurt you, yeah.
Do the seats fill up pretty quickly? Like, are there a lot of people that attend it? To where you have to kind of get here a little bit earlier?
No, not for that one. Not for that particular one, I don't think so.
Okay.
Okay, we do the science lecture series, which you may be aware of that too. So those are usually better attended because it's campus wide.
Okay.
So this is more just biology mostly, and not all the biology classes. I mean, they're all invited, but they're not all going to come. You know. So anyway, you might find it kind of interesting. Okay, so yes.
And generally, how long is the lecture?
It's be an hour, plus or minus. It might be sooner. I don't really know, you know. So we gave the speaker, we said, you know, about an hour, and then have a question and answer session. So, okay. Anyway, I don't think you need to get there early and, you know, it's gonna sell out or something. That would be nice. Okay, so what I need you to do, guys, is I need you to get out your selective and differential medias lab. We're going to kind of go through the results. I just want to, because we kind of rushed that day, because we had a quiz, I want to make sure we're all up to speed. Okay. So get those out, get your discussion questions out, and I'll pull this computer and wake it up. Selective and differential media lab, is that 70? Hey, 70, thank you. So you want to pull that open. We're going to kind of go through the results that you should have gotten. And we'll talk a little bit about what they mean and how you might answer discussion questions. We're going to get caught up for 40 minutes or something. Just to make sure everyone is on the right page. Because this was a really important lab. And we only had to rush it a little bit because of the quiz. And then how about the special questions? Okay, so as it turns out, the lecture exam covers, the one that's in, you know, a week and change, covers this topic, selective and differential. So you'll notice there's overlap between lecture and lab. They don't always go, you know, at the same time, but you should definitely know this stuff. So when I'm reviewing here today for lab, I'm also reviewing for the lecture exam. Okay, so the selective and differential media lab, we actually did a few plates with your unknowns. So we'll talk about the unknowns a little bit later. But we also showed you some controls. Okay? So hopefully in your notebook you should have some circles. You should have your unknown mixture of bacteria on salt. And very few of you had growth. But let's see if you kind of remember what that means. If you had growth of your unknown, what do you know about your unknown, or at least one of your unknowns? It could be staph. It could, I hear somebody else saying that it could be halotolerant, which is a little more general term. It means it tolerates salt, which is relatively rare. So if you had growth on salt, it was a halotolerant organism, maybe staph, but also maybe Corynebacterium, Bacillus, and Mycobacterium. All three of those and staph are gram positive. You had a 50-50 chance. Staph, you should always remember staph is gram positive. I mean, that's kind of our example all the time. So if you got something to grow on here, you know it's your gram positive, okay? So for comparison, we showed you 4 control organisms. Which of the 4 control organisms, you look in your drawings there, which of the 4 control organisms grew? Staph, epidermidis, lo and behold, sure enough, okay? So you should label that, the one that grew, staph epidermidis, you should have these labeled with the names of the organisms, and you should say positive for growth. The other 3 are all gram-negative bacteria. E. coli, Enterobacter, Salmonella, they're all gram-negatives. None of them grew, okay? So you can say negative for growth, negative for growth, negative for growth. That's what you should have in your notebook. Don't forget the labeling. So I'm kind of emphasizing labeling and things that I look for when I look at your notebooks, okay? Then we also did another plate, only this plate was a McConkey plate. So the McConkey plate is both selective and differential. So what does it select for? What types of organisms will grow? Gram-negative, gram-negative. So remember that the gram-negatives have that extra outer membrane, and it makes them relatively resistant to chemicals, okay? So the gram-negatives will grow on McConkey, and then additionally, it also differentiates what characteristic? Lactose fermentation, okay? So in the agar, there is lactose, and if your organism in growing can take the lactose and ferment it, it will produce acids. And those acids are going to do what to the pH of the media? More acid, pH is going to go down. So as the pH goes down, you see a color change in the media. A color change. So as the plate itself starts out kind of a light pinky color, and as the bacteria grow, if they ferment lactose, they will darken the media around them. They'll have pink colonies if they ferment lactose. So if they ferment lactose, acids will be produced. The acids decrease the pH, and you can see it with a pink color change on the media, okay? So, how many of you had growth on your McConkeys? I think all of you had growth on your McConkeys. I believe it in the morning class, maybe not in the afternoon, but in the morning class, everyone got something to grow on McConkey. So that tells you that was a gram-negative. And additionally, you should have looked to see if the colonies looked relatively pink or sort of colorless, or maybe even sort of a brown color. So if you noted that, you might want to write it up here. Growth positive, lactose fermentation positive, or something like that next to that. That's a note to you to pay attention to that. It turns out that's an important characteristic. Additionally, we ran 4 control organisms, the same 4 that were on the salt, only again, make sure you label their names, and you can abbreviate now. As long as you've written it somewhere, you can start abbreviating: S. epidermidis, S. typhimurium, E. E. aerogenes or cloacae, I forget which one. Aerogenes, thank you, and E. coli. So notice why you have to abbreviate. Because it's not Escherichia aerogenes, it's Enterobacter aerogenes. You see what I'm saying? You have that's the point of that abbreviation thing. Okay, so of those 4 organisms, one of them didn't grow. Which one that didn't grow? Staph epidermidis. Okay, so you can just write no growth. Because it didn't grow, you can't tell if it ferments lactose or not. So that means a lot of times students think if it didn't grow, then it can't ferment lactose. Actually, staph can ferment lactose. But if it's not growing, you can't see the pH change. You can't see it. So just say no growth for your staph. Then of the other 3, 2 of them fermented lactose, and 1 of them did not. Which one had the sort of colorless, lactose negative colonies? Salmonella. So salmonella, you should label it. I don't care how you drew it on here, but just make sure you label salmonella. It grew, growth was positive, but no lactose fermentation. And as it turns out, guys, that's an important characteristic in the clinical setting. So even though these kind of plates were developed, 100 years ago, they literally still run these kind of things in the clinical setting. So if someone, for example, has a diarrheal syndrome that doesn't go away and makes them really, really, really sick, and they end up either going to the hospital or in urgent care, and they can't figure out what's going wrong with them, poop in a tube, sorry, they will run it on either a McConkey or something like it. They may run other media too, but just this is for an example here. So in that poop sample, guys, there's a million things. Most of the normal flora found in poop are gram-negative, so they'll grow mixtures, okay? It's not just one thing, there's a lot of stuff in there. So how do you pull out the pathogen from all the normal stuff? Well, that's what this sort of media allows you to do. So you got 100 things in there, and the gram-positives won't grow, only the gram-negatives. And most of the normal flora, listen carefully, this is a discussion question, guys. Most of the normal flora are lactose fermenters. Most normal flora in stool are lactose fermenters. So E. coli, Enterobacter, Klebsiella, etc., all of those are lactose fermenters. But if you get a stool sample from somebody who has a diarrheal syndrome and a fever syndrome and is getting sicker and sicker, and they test their stool, and if they have in that mixture of things in their stool, they have some non-lactose fermenting colonies, some colonies that are not fermenting lactose, they start thinking, oh, that's salmonella. Because salmonella does not ferment lactose. Do you get it? So most normal flora ferment lactose, yes. Most of the pathogens, including salmonella and another worse one called Shigella, don't ferment lactose. So you can see how this kind of plate might be used in a clinical setting to pull the pathogen out from the mixture of normal flora. Because they're always kind of have this problem in the clinical setting. You don't have one one species in a stool sample. You get a ton of stuff in there, and you basically have to fish out the pathogen from all the normal flora, and that's one of the reasons for using McConkey or a similar one. The one I talked about in lecture is called EMB, eosin methylene blue. It does the same thing. It selects for gram-negative, and it shows differential characteristics of lactose fermentation. It's a little more expensive, so we don't use it as much. We'll use it later on this semester. Okay, so hopefully you can label the controls with growth positive, lactose negative, or growth positive, lactose positive. So do you get it? And then you're supposed to kind of compare your unknown to that. Don't worry if you didn't figure it out for your unknown. We're going to do another tube test that will confirm the lactose result on your unknown. Bless you. Okay, and then finally, we did some blood agar. So if you remember, blood agar is kind of expensive, so we don't use it routinely on the unknowns. Later, a couple of weeks from now or a month or so from now, some of you who think you have staph or enterococcus, which is kind of like streptococcus, you might be given a blood agar plate. But again, we don't give them to everyone because most people don't need them. So the blood agar plate is not selective, not selective. Everything, not everything, but most bacteria that can be cultured in the lab will grow on blood agar because it's super rich. And it has sheep red blood cells. And if you remember, they were bright red, at least initially, bright red, like, arterial blood. So then we put some bacteria on it. So One of the control plates had 3 species of staph on it. And those are by the way, we're introducing, in these controls, these are bacteria that are used as unknowns. Okay, so that's why you kind of want to pay attention to those. But the 3 species of staph that are given as unknowns in the class are Staph epidermidis, Staph saprophyticus, and Staph aureus. So remember, for these blood plates, you're supposed to hold them up to the light. So look in your results there, and hopefully you drew somehow which one of the 3 staph, which one was lysing the red blood cells? Staph aureus, thank you. Staph aureus was lysing the red blood cells. And in fact, Staph aureus is said to be beta hemolytic. Beta hemolytic is complete lysis. So you can see where the bacteria are growing, and then there was this little halo thing around them. Let's see if I have a picture here, kind of show you. So you can see here's a Staph aureus. There was like a halo around it where it was completely lysed in that place. So that's called beta hemolysis. So Staph aureus, we call it beta hemolytic. You should label your plate as such. And the other 2 are growing but not really doing anything. That means the other 2 staph. So what do we call that? Gamma. Gamma. Looks like, like a fish thing on its nose. Okay? So those are called gamma. So you got to label your plates. Beta hemolytic, gamma, gamma. Okay. So from lecture, do you guys remember there's another bacterium that is notably beta hemolytic, and it causes strep throat. Do you guys remember this from lecture? So the one that causes strep throat is called Streptococcus. Streptococcus pyogenes. So Streptococcus pyogenes, pyo means fire generating. That's literally what that name means. It means like fever generating. Okay, so Streptococcus pyogenes causes strep throat. It's the one of the flesh-eating bacteria. It's a really important pathogen. And if someone has strep throat, usually when you go to the doctor, they do a rapid test. So the rapid test, they can tell you, pretty much right away after they do this, add the reagents, blah, 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 they come back and go, yep, that's strep throat. But sometimes it is too early in the infection, for example, where they get a false negative, but the doctor's like looking in your throat going, yeah, that's red and raw, that really looks like strep throat. So the doctor will usually go ahead and give you or prescribe an antibiotic. But they usually take a throat swab and run this old school as a backup. And this old school method, like literally, uses blood agar. So think about that. In your mouth, you have a million things. Not quite as bad as poop, but you have a different garden in your mouth. But it has a whole mixture of things. You have normal flora and you have pathogens. If you have strep throat, you're going to have strep pyogenes, and when they do that throat swab, they're going to find beta hemolytic colonies on the blood agar plate. Beta hemolytic colonies on a blood agar plate from a throat indicates strep throat. There's hardly anything else that should be in your mouth that is beta hemolytic. Okay, so most of the normal flora in your mouth, and remember we learned that in the beginning of the semester, there's a lot of normal flora in your mouth. Okay, most of those are gamma, or there's a few of them that are alpha, which is partial hemolysis. But if they see beta hemolytic colonies, they're not thinking Staph aureus. They're thinking Streptococcus pyogenes. Okay? So that one's such a bad guy that we don't culture it for lab. We don't give it to students as unknowns for obvious reasons. So Staph aureus, sorry, a lot of you have it in your nose, in your armpit, in your groin area. So we can't protect you from this one. Okay? So we do give this as an unknown, but it is a pathogen. So you always, that's why I'm trying to get you, please don't play with your cell phones in class. Okay? Please don't play with your cell phones. So, because you don't want to take something home in huge numbers, those billions of bacteria that we saw, remember, from our counting viable bacteria lab. So anyway, you don't want to do that, okay? So the two on the other plate were from the genus Streptococcus, is kind of undergoing a name change currently, like literally in the past 5 years. So on your notebook, it should say Enterococcus, right? Does it say Enterococcus? Okay, just know Enterococcus is formerly known as, not Prince, but formerly known as Streptococcus. So that means it's undergoing a name change because they're trying to separate this guy out from all the other ones. Okay, so bottom line is the one that was showing some hemolysis, partial hemolysis, we call that alpha, should have been, and look on your notes and make sure you got it, it should be Enterococcus durans. Did you guys see that? Enterococcus durans was alpha hemolytic. Okay, so good. Okay, so that means that most of you saw what you should have seen. Okay, so knowing that, do you think you can answer the discussion questions? Look at the discussion questions, guys. We didn't give you time, but we're going to give you some time now. Look over the discussion questions. First discussion question. I don't know if you guys can read this, but by the way, you probably should have this actually like printed. I see people trying to read this like on their cell phone in lab, and it's like A, you shouldn't be playing with your cell phone in lab, and B, can't really read it very well. Anyway, I know you guys are young eyeballs, but I see students, trying to read it on their cell phone. Okay, so, why is hemolysis considered a virulence factor in vivo? Okay, so in that question, I'm bringing out, or sort of introducing you to some terminology. So who's heard of the term in vitro fertilization? You guys have heard that. In vitro fertilization, which essentially means artificial fertilization. The actual, if you were to translate it, in vitro means in glass, in a petri dish fertilization. In vivo means in life. And that kind of fertilization is a lot more fun. Anyway, in vivo, it means in a real setting, okay? So this question says, why is hemolysis considered a virulence factor in vivo? That means in patients, not in a petri dish, but in a patient's body. Well, you gotta know what a virulence factor is. A virulence factor is anything that harms the host or interferes with the host immune function. I'll say that again. You need to know it for lecture, and this is going to come up a number of times. A virulence factor is something that harms the host in some way or interferes with the immune function of the host. So guys, you know that most bacteria don't hurt you, right? The ones that live on and in you, they don't hurt you. Why not? Because they don't produce virulence factors. That means they are harmless. But there are some notable virulent bacteria that cause harm. Why do they cause harm? How do they cause harm? Well, Streptococcus pyogenes lyses red blood cells. Not just in a petri dish, guys. In life, in you. They lyse your cells. They break open your cells. Okay? And by the way, they don't just lyse red blood cells. That's what you see on the plate. But in real life, they lyse white blood cells too. Okay, so very, very virulent organism. We'll talk about some more of their virulence factors later in the semester. Okay? So, I think you should be able to answer number 2, since we just went over it. What makes MAC selective, differential? That's in the lab write-up. Describe how MAC, McConkey, could be used in the clinical setting, differentiating normal flora from pathogen in stool samples. I think I just went over that. Did you guys hear that? Talked about that. And then the last one, why does it make sense that staff are halo tolerant? They live on the skin. The skin is salty, you know, transiently, but it is salty. It's way saltier than the inside of your body because you produce sweat that is salty. Does that make sense? So instead of waiting to do the discussion questions some other day, 2 weeks from now, 3 weeks from now, 4 weeks from now, do it now. Like literally, right now. If you haven't already done them yet, I don't think you had time to do them. So let's just spend literally 15 minutes going through that now. Okay, so don't look at me. You should be able to answer those. Try to answer in complete sentences. Try to flesh it out a little bit. I'm gonna get the stuff for the next labs, but let's get that done. Help each other. You can talk to each other.